Hello, and thank you for coming to my poster. My name is Constantin Schneider, and today I'm going to present some recent work that we've been doing in the Oxford Protein Informatics group in collaboration with researchers at AstraZeneca on deep learning methods for structure-based virtual screening of antibodies. To just briefly set the scene, our work aims at supplementing the early stage of antibody drug discovery, which is mainly conducted using experimental high throughput screens against antibody libraries. Our work then aims to supplement this stage using computational structure-based high throughput screening tools. To this end, we have developed DLAB, a virtual screening pipeline combining antibody modeling, rigid body docking against antigen targets of interest, as well as convolutional neural network tools, both for rescoring and quality assessment of docking poses, as well as classifying antibody antigen pairings into binding or non-binding pairings. And I'll quickly walk you through this pipeline now. The first step is obviously modeling an antibody of interest from its sequence. To this end, we use the bodybuilder modeling tool, which is both fast and able to create accurate models of antibodies of interest. We then use these antibody models in order to generate docking poses against antigens of interest. To this end, we use the fast rigid body docking tool ZDock. These docking poses are then reassessed using the convolutional neural network model DLAB-RE. We term to DLAB-RE for rescoring, which re-ranks the ZDock output. We are able to use a convolutional neural network here to re-rank poses because after generating the poses using the docking program, we convert them into three-dimensional grids of atom type densities using the libmol grid Python package, which obviously enables us to very readily use them as inputs into convolutional neural network architectures. Using this input format, we were able to generate the DLAB-RE model, which, as you can see here, is able to improve the ranking of docking poses generated by ZDock over native ZDock, generating ranking near-native poses more accurately at the top of the pose ranking. And in some cases, as you can see here, this is indeed quite significant, as there are cases in which the DLAB-RE tool does indeed promote very good poses to the top of the ranking, which were missed by the ZDock scoring function as well. And secondly, we use the DLAB-RE scoring output to perform quality assessment on the ZDock runs. By using the highest score associated to any pose of a particular antibody-antigen pairing, we determine whether we believe that any particular antibody-antigen pairing was well docked, if near-native poses have been produced in the first place. And by discarding antibody-antigen pairings for which low scores have been assigned to all poses generated by ZDock, we can indeed identify those pairings which have been well docked and well ranked. Using the output from the DLAB-RE, we then make a classification into either binder or non-binder, i.e. do we believe that a particular antibody-antigen pairing is indeed a binding pairing. For this purpose, we use a second convolutional neural network model, DLAB-VS for virtual screening, which uses as its input poses of antibody against antigen and then creates a binary classification score. We assess this tool by ranking a cognate antibody against its antigen, as well as 50 non-binding, non-cognate antibodies against the same antigen, and attempting to retrieve the correct binder from this background. And indeed, our model is able to enrich the correct binder at the top of these rankings. And using the DLAB output to identify antigen targets on which the model performs well, we're able to further improve the model's performance. We then went on to test the model on a realistic use case in which a set of binders to the SARS-CoV-1 receptor binding domain had to be retrieved from a set of 300 length matched non-binders. And indeed, as you can see here, both before and after thresholding using the DLAB scores, correct binders were promoted to the top of the ranking, demonstrating the potential utility of this tool. We therefore believe that we've demonstrated that structure-based deep learning tools are very applicable to antibody virtual screening, and in the future, such methods like DLAB will continue to improve as more structures of antibody-antigen complexes are generated, as well as structure prediction of antibodies and fast docking methods are continuously improved. If you want to learn more, our preprint is out on the bioRxiv. The DLAB source code as well as pre-trained models are available on GitHub, and I will be talking about this in a bit more detail on Tuesday, so please join us there if you want to know more. Thank you very much.